Takeshi Iwata. I'm from the National Institute of Sensory Organs at the National Hospital Organization of Tokyo Medical Center.
Tell us about the research you presented.
So I presented some of the work that we are currently working as a Japanese eye genetic consortiums and the global eye genetic consortiums about AMD, how we collected samples of AMD patients throughout the world and analyzed the cause of this diseases. So we have worked on GWAS studies, the whole genome association studies that link to the locus of the chromosome 10, which two genes, ARMS2 and HTR A1, is located. A number of people have worked which gene is actually responsible for this AMD, but our research results show that this HTR A1 could be one of the targets for therapies. And our data shows that the sequence difference between the normal person and the AMD patients are located immediately after this ARMS2 genes. There are 250 base pair sort of insertions and deletions in this region that triggers the induction of the transcription of the HTRA1s. So we have worked on this region, why this 250 base pair of insertion deletions will affect the HTRA1 transcriptions. And also, if you induce the transcription of HTRA1 in mice, would you get a human AMD-like phenotype in the mice? Nobody has actually showed that kind of phenotype. So we simply made the transgenic mice using a chicken actin promoter, and after 1 year we were able to get in some of the mice choroidal neovascularizations in these mice. Number of people have tried inducing HTRA1 in RP cells, but they were not able to get this choroidal neovascularizations. Rather, they were able to get this differences in the choroid rather than the retinal vasculature. So I think we overexpressing HTRA1 in the entire body rather than just specifically in the RP, or the work we did for photoreceptors, specifically inducing in photoreceptor will not give a CNV in mice, but only when you overexpress in the entire body, then you'll get CNV. That sort of suggests that in humans, HTRA1 is not specifically expressed in the eye, it's expressed in kidney, livers, and all other parts of the. So maybe they are induced in the whole entire body, and because the HTRA1 is a secreted serine protease, it can be secreted out from these tissues and come to the choroid to destroy the membranes of the blood membranes and other choroidal components.
Are you working on suppressing certain genes as well?
So there are a few companies working to inhibit the HTRA1. I don't think you need to inhibit completely. You just want to reduce the amount to a normal level so that you will prevent on sort of tipping over this amount that triggers the AMD. So I'm not working directly with the drugs, but there are companies working on this, and then we're supporting these animal models to them to get their data.
In your international consortium, have you observed a specific area where these genes are more common?
AMD International Consortium has shown, collecting DNA from many parts of the world, that for chromosome 10 HTRA1 locus is quite stable. It has the most lowest p-value for AMD, which means that it's highly associated, and also the odd ratios. Having these mutations would cause these diseases is over 5 in many of the ethnic groups, which is almost like a Mendelian diseases. Yeah.
Are these genes more associated with the wet or dry form, or just AMD in general?
We have, in the Japanese population, worked on wet type of AMD because majority of the AMD in Japanese is wet. But many other countries are working on other dry types. They're also showing association. So I think this HTR A1 theory does not only apply to the CNV, but also for geographic atrophy as well. And we have not really looked into that yet, but we have given our mice to many research around the world, and they're also working on these dry type AMD.
What are the next steps in this line of research?
We want to know how this sequence differences will induce the transcription of HTR A1. So we have identified a transcription factor that's specifically been isolated from this insertion deletions regions. So we are going to study this proteins, how it's activated. I'm sure this is not one single protein, it's a complex of proteins. And I think these complex of proteins can be influenced by the environmental factors. This is not a Mendelian disease, so if you have a mutation, that doesn't mean that you have the AMD. I think there is a factor within these complex proteins that can be influenced by the environmental factor. The reason I'm saying this is that the transgenic mice we made, all mice doesn't get CNV. Only 20% of mice get CNV, which means they are living in the same segment of the room, they're eating the same, breathing the same air and same water, but still not all the mice get CNV. Why is it? We want to know. There are some other mechanisms that influence the CNV or not CNV.
What are the environmental factors you are looking at that causes AMD?
We have not really looked into the smoking, but I think maybe the light, because if the cage is on the upper part of the cage, it's closer to the light. And the CNV that we see in these mice are near the optic nerve, which means that where the light is focused on. So maybe light, maybe a factor. We have not really extensively worked on this, but I think we should work on it.
How do you see this study translating into patient care?
Because there are millions of patients around the world, and especially in the Caucasian is #1 cause for blindness. So I think many people are waiting for this, and many of the people who are treated with the anti-VEGF, not all of them patients is cured by this. So I think we need a fundamental drug that will rescue these patients. I think HTR A1 theory is based on the GWAS studies built up by in vitro studies and also animal studies. I think there is enough back of information to build a very strong treatment through HTR A1.
Are there pharmaceutical companies currently working on a gene-targeting therapy for AMD?
I think there are some companies that are preparing for clinical trials, and because there are many serine proteases, making a specific inhibitor for this HTR A1 is going to be very challenging, I think.
Where can someone find more information about your research and the work you're doing?
I have a website. You can log into iwatalab.org, iwatalab.org, lab.org.